All right. So today we're going to start a lesson that we'll likely finish tomorrow. We are currently coming at you live from A7 here in downtown. If you're new to our channel, please subscribe. All right. So, like usual, you'll find this on Schoology if you want to go back and rewatch it. Okay. I should start running ads.
You should.
Just like a side hustle, just have ads on my YouTube channel. Okay. So, what I want to accomplish on this lesson are two things. The first thing, I want to do a brief recap of cell communication, but then I want to spend most of my time talking about signal transduction today, as well as some examples tomorrow. Okay. So, the first thing here, we mentioned cell communication falls into one of four categories, right? Those four categories are, first one, autocrine. And by autocrine, like the name implies, this is communication that happens from self to self communication. In other words, a cell is releasing signals that are received by itself. So an example of this, we have a cell here. This cell has a receptor. And will release a signaling molecule that binds to that receptor. Okay. So this is our ligand. This is our receptor. And this brings about some kind of response. Now, one of the most well-known examples of this are tumor cells. So a lot of times, a tumor cell will release growth signals to itself to promote division. Its own cell division. It'll continue to release that signal over and over and over so that that cell can divide over and over and over again. Okay, so autocrine, self-to-self signaling. Okay. Second type. Something called juxtacrine. You also might see the term synaptic, because this type is usually associated with synapses. But wait, I'm sorry, that's a mistake. Go ahead and cross that out. Not a mistake. That's paracrine. So juxtacrine, what we're referring to here is cell-to-cell contact signaling. cell-to-cell contact. So there are two different cells involved here, but they have to make direct contact in order to signal with each other. And there are two kind of hallmark examples of juxtacrine signaling. The first one occurs in plant cells. So these are two plant cells. And plant cells have these structures connecting cells called plasmodesmata. So those structures, plasmodesmata, their function is to allow signaling to occur between plant cells. Plasmodesmata. And these are our plant cells. That has a lot to do with plant cells have cell walls, and oftentimes it's hard to get signals through there, so they have these plasmodesmata to help facilitate that. Example 2, which Mr. Anderson went over in the cell communication video, is called, or are called, APCs, or antigen presenting cells. And this is in the context of the immune system. So what happens here, and you can go back, I'll make a little note here, look at the Bozeman video. But what happens here is that you have cells within your body whose job it is to defend against foreign infection or antigens. And so what you typically have will be a cell such as a macrophage. And the macrophage, its main job is to literally eat things. It'll go around and will ingest, let's make this red piece. Let's say this is a virus. Okay. So it'll go and it'll ingest this virus and it'll capture it. In a membrane-bound sac. In a process similar to endocytosis, it's called phagocytosis. And what the purpose of this is, it'll capture it. And then with the aid of some lysosomes, who will deposit some enzymes, it'll break down that virus into a bunch of small little pieces. And what's pretty cool about this is not only will it break it down, but it'll take some of those broken down pieces, so little small pieces, and it will present pieces of that on a little cell receptor on its outside edge, on its membrane. Right? So it will digest it and it will present these little pieces, and we call those little pieces antigens, on its receptor. And this receptor has a specific name. It's called an MHC class 2 receptor. If you go into immunology, you will know a bunch about this, but its job is to just present the antigen. It's a protein that does that. And then you have this little buddy called a helper T cell that'll come by with its own receptor to recognize that antigen. Right? And once it does that, it can actually go to B cells. It'll activate B cells to make antibodies. And it'll attract killer T cells, which I think is an epic name, to assist the macrophage. Right, so this helper T cell, it's also known as a CD4 positive cell. Its job is to recognize that antigen being presented and then go do something about it. Okay, so this cell-to-cell contact right here, that is juxtacrine signaling. Okay? It's a hallmark of juxtacrine signaling. Okay. Again, if you want to learn more about that, go back to the Bozeman video, but also come talk to me, I'll talk to you about it. Okay. So that's juxtacrine. Other type here. This is paracrine or synaptic. Paracrine or synaptic. And what we're referring to here is close range signaling. So not direct contact, but close range. The two major examples, one is quorum sensing in bacteria. So hopefully you watched the clip over the break. Bacteria have this mechanism of releasing signals. This is bacteria. This is the signal. They release this signal and they have little receptors to attach to these signals. And the major purpose is that they're able to measure their own population density. In other words, are there enough of us to be able to take down this host that we're infecting? And if so, then they'll start producing the toxins, the things that make them virulent. But if not, they'll kind of hang low until they're ready. Right? Again, there's a clip that I had you watch over the break about quorum sensing. Pretty awesome stuff. But that's one example of paracrine. The other example, as the name implies, is something called a synapse. All right, this is the gap between neurons. So if you have neuron 1 and neuron 2, you'll have some receptors that bind to these neurotransmitters released from neuron 1. This is N1. This is N2. Okay. This is our synapse right in the middle. Okay, those neurotransmitters crossing the synapse are received by the next signal and cause some response. Okay. What's that last type? So we talked about 3 types here. There's one more, J.E. What is it? So it's an endocrine system is responsible for the fourth type. So we've got juxtacrine, paracrine, autocrine, and endocrine. So, endocrine here. So endocrine. This is our far range signaling, where we typically will use a gland to release a signal, typically a hormone, into the bloodstream to reach far away cells. Okay. Now, we're going to talk about a couple different examples today and tomorrow, but these glands, so some common glands that you will see include the pancreas, the ovaries or the testes, the pituitary, the hypothalamus, and the thyroid. Right, all of these are glands that release hormones into the blood, or release some kind of signaling into the blood. Okay, and we'll end with this. Some common hormones. Common hormones include insulin or glucagon. We also have epinephrine, or it's also called adrenaline. We have testosterone, or estrogen, and human growth hormone, or HGH. So different types of hormones, okay? They all do different things, but those are some key examples. Okay. So tomorrow, we're going to talk a lot about cell signaling pathways. We'll talk about a couple different classic examples. We'll go over GPCRs, so G-coupled protein receptors, that you saw over the weekend. Once you have that, you can put your notebook away. Again, test corrections start today at the school, and they'll go through Thursday. So make sure you come talk to me if any of those times don't work for you.